My name is Jennifer Doudna. I'm a professor at the University of California, Berkeley, and a Howard Hughes Medical Institute investigator. My research involves studying how bacteria fight viral infection. And in the course of that work, which was a project to discover fundamentally an aspect of science that nobody had investigated previously, we found out that a protein called Cas9, which is part of the CRISPR bacterial immune system, is an enzyme that can find and cut DNA sequences in cells. It's a molecule shown in this model, in the white part of the model, that holds on to double-stranded DNA and makes a cut just like a pair of scissors. And it can be programmed by bacteria or by scientists to find a particular DNA sequence and make a break. And that's important because when that occurs in an animal or plant cell, the cell will repair the DNA and in the process make a change to the DNA sequence. And this has been the advent of a very exciting technology for genome editing, a way that we can alter DNA in cells precisely and simply using this tool in ways that in the future are likely to lead to cures for genetic diseases, as well as lots of new insights into the fundamental aspects of genetics in biology. CRISPR-based gene editing gives scientists a tool for manipulating the DNA in cells that's unprecedented. We can now understand the fundamental genetics of any organism at a level that previously was impossible, really even to imagine. Lots of people think about the use of CRISPR and similar technologies to cure genetic disease in humans. I think one of the exciting, profound advances that will happen in the future actually has to do with understanding plants and figuring out how plants live in their environment, why plants have the traits that they do. Plant genomes are typically much, much larger than the human genome. And with the CRISPR tools, we now can manipulate plant genomes to understand the content of that DNA and how the traits in plants are encoded in DNA and can be manipulated now in the laboratory using this kind of technology. So I think we're going to see a revolution in understanding our natural world by the use of CRISPR-based genome editing.